So what are some of the things that I want to keep you in mind? So look at these charts, right? Green, blue. Green, bacteria. Blue, viruses. Notice the bacteria are living organisms. The viruses are not living organisms. They only work and function when they're inside other cells, right? And when you look at how you compare across them in different sizes, what you'll see is that the bacteria and viruses have very different sizes, orders of magnitude, right? So you start off with the little genetic material, but you can see you can go from a virus up to a bacteria, then up to a red blood cell, then up to a human cell. So it gives you an idea of how very small these are, and yet we're working with something very small yet very powerful. When we talk about infections, right, and people say, okay, I want to know about infections, keep this in mind. Not all infections are viruses, not all are bacteria, and that's why a lot of you know that when you have colds and flus and you come see me or you call the office, I will say, please don't take an antibiotic, because the majority of these things are viruses for which antibiotics are useless, and that's why you don't want to take them. When we're talking specifically about the Sars Cov2, it's a beta coronavirus, and this comes from other SARS. There was one previously, and other bat coronaviruses. Now look. This is the largest RNA virus that there is that we know that infects humans. 10 to 30% of common colds are due to coronavirus. Please keep that in mind. This is an important statistic because you need to know that the odds are that at some point you've already had a coronavirus infection. It's also important because when we start talking about certain things like reinfection, like antibody testing, you could start to understand where there would be a scenario where there's an overlap in the testing between a regular RNA virus that's causing the common cold versus this RNA virus that's causing a more severe infection, right? It's called corona because it looks like the sun, and SARS stands for severe acute respiratory syndrome. MERS, which is a closely related cousin, you may have heard about. These both SARS came from the same area of China. Now, the whole thing about, okay, there's a medical research lab there, and this is very unusual having both there, and bats are not endemic there, but there's also a possibility in animal hosts might have been involved. I don't think we're ever going to get that resolved, so don't plan on getting a resolution. You can't get total transparency in certain types of governments and such, so it's really hard to know, but that's where that whole controversy comes from, right? But when we look at the SARS outbreak in the past, now look at this number, look. In 2003, 2004, it only lasted 9 months, and look at that mortality rate, 10%. This is gonna come up again when we talk about other things, right? Look at the MERS. The MERS was an epidemic. Notice this, look at the change in terms. It's an epidemic. It's not a pandemic. Why? Because it had such high mortality that it couldn't spread as easily, because quite frankly, anything it came in contact with, it was knocking down and killing, right? So that's why it never spread very far, because it was so severe, right? And then we call this coronavirus disease in 2019 COVID-19. That's where that term comes from for everybody, okay? Next, is reinfection possible? Okay, maybe. There's no human reinfection that has been fully confirmed, but there are now anecdotal reports. This is very important. In the past, when I was looking at this literature, when a lot of people were looking at this science, they're saying, okay, well, maybe some of those people didn't really have the full coronavirus. Maybe they just had kind of a weird exposure and only had some brief symptoms. Maybe they were showing antibodies from an old coronavirus infection from the common cold. Maybe that they never really cleared up very well, that they had the infection, never really went away. But now there's been some case reports that I think are very credible that suggest that it is possible for reinfection. How common is that going to be? How likely is it going to be? It's going to depend on a lot of factors, right? So for example, it may depend on somebody's immune system. It may depend on how much this virus has mutated. So let me just give you for some numbers, right? Influenza. Influenza mutates about 50 times a year, which is why you know that we give a flu shot every fall. Because if this virus changes so much, then we have to give a new flu shot in the fall to kind of cover those changes. So all of a sudden you start saying, well, wait. What if we get a coronavirus vaccine one day? Will we also have to change that? It's possible. It's possible that if we're lucky to get a vaccine, it may be that if this mutates enough that you might have to do that vaccine all over again every year to try to get at that treatment, right? Now, these concepts that you may hear about, read about, so I'm just saying it once, you've heard it once, called antigenic drift, antigenic shift. These are mutations. This is where virus changes. But remember, not all mutations are bad, right? We've already talked about the fact that you've got mutations where there were some of these viral particles which cause the common cold. They're not bad. They're not severe. They're ones that we easily clear up. So you can start to see how there can be a difference in some of these where it may mutate, but it's not necessarily a bad mutation. For example, right now, there's some good data that's suggesting that in Europe, they've already got a little bit different of a mutation there. That the European strain is more virulent, which means it more easily infects people. On the other hand, the mutation seems to be less lethal. So not every mutation is bad, and that can change over time, right? Okay. Now, when you look at an electron microscope, this is what they look like, highlighted in blue. Here's a little model of what this virus looks like. I know this gets a little bit confusing, but just to let you see kind of what's already known, what's already been worked out about some of these different things. Now, here's a simple model. I bring this up to kind of highlight that for a reason. Look here. You see the E protein, envelope protein, S, spike protein, M, matrix protein. So you do these different types of proteins that are all different parts of the coronavirus, right? And these are what are used to identify the type of virus. These are what are used to identify mutations. These are what are used for vaccine development, right? And so that's why these become very important. So the S protein, the spike protein, is these most specific, which means that if you have a blood test that's looking for the antibodies, the antibody blood test will be more reliable if it's based on the spike protein, the S protein.